Hello and welcome back to our lecture dealing with the adaptive immune system. In this last video we are going to be dealing with the link between the innate and adaptive immune system. We will talk a bit about vaccines, and lastly we will learn what happens if the immune system or immune response goes wrong. So again, we have learned that the innate immune response is the first and second line of defense. If this fails, this triggers the activation of the adaptive immune system. But how are those two systems actually linked? And the answer is simply by macrophages and dendritic cells. Those cells are able to communicate with the adaptive immune system cells, and they can trigger it. So how does it work? Again, if you remember what I told you last week, macrophages and dendritic cells do express very interesting and specific receptors on the surface, referred to as pattern recognition receptors or toll-like receptors. In the human body there is a huge variety of those receptors, and those receptors are able to recognize very different components of pathogens. They can recognize pathogen-derived lipoproteins, CpG-rich DNA, which is less common in eukaryotes. They can recognize short RNA molecules that are single-stranded. They can recognize double-stranded RNA. They can recognize bacterial lipopolysaccharides and other components of microorganisms. Binding of those pathogen-derived patterns or molecules to those receptors triggers activation of the so-called NF-κB pathway, which results in the transcription of immunomodulatory genes and also cytokines. So, when a pathogen-derived molecule is recognized by those pattern recognition receptors or TLRs present on the surface of macrophages and dendritic cells, this leads not only to pathogen phagocytosis and elimination, but also to secretion of cytokines. In case of dendritic cells, those cells can actively present antigens derived from the pathogens on the surface to T cells. And this is how those two systems are actually bridged. And the key step here is delivery of antigens to T cells by those phagocytes. And this is something you should keep in mind. So in the antigen recognition phase, dendritic cells, sometimes also macrophages, present the antigen to naive undifferentiated B cells and naive undifferentiated T cells. This leads to clonal expansion of both of those cell types. Then those cells differentiate, and the mature B cells or plasma cells start producing antibodies. In case of T cells, active effector T lymphocytes or T cells are formed, and they actively eliminate infected cells, and this is the antigen elimination phase. Afterwards, after the antigens are actively removed and the infection is actually dealt with, those cells undergo programmed cell death or apoptosis. However, some of B cells and T cells are not active during this phase because they differentiate into so-called memory cells, into B memory cells and T memory cells. Those cells can persist for quite a long time after the infection, and those cells are the driving force in case of the fast and strong adaptive immune response if the body encounters a certain pathogen for a second time. And as I mentioned before, those memory cells are responsible for the function of vaccines. So, vaccination, and this is something I hope that you looked at in detail in the lecture delivered by Dr. Black, is a process of induction of immunity to a pathogen by deliberate injection of a weakened, modified or related form of the pathogen which is not longer pathogenic. And vaccines are incredibly important for you as future pharmacists and future physicians associates, because you should be vaccination advocates. Vaccines are incredibly important for the health systems around the globe, and they are especially important during the ongoing COVID pandemic. So, how do vaccines work? In simple terms, the body is exposed to a weakened form of the pathogen. This leads to a primary antibody response and, more importantly, formation of memory cells. If now the body encounters the same pathogen again, in case of a natural infection, this leads to a strong and very fast secondary antibody response, because the body acquired immunity, which was driven by application of the weakened form of the pathogen before. So, in terms of vaccines, there are many different platforms how vaccines can actually be delivered, and we have inactivated vaccines. Example of approved vaccines are flu vaccine, polio or rabies vaccine. We can have subunit vaccines, and examples are hepatitis vaccine. We can have virus-like particles, viral vector vaccines, and the more modern approach is to deliver vaccination via the use of DNA or RNA. And this is actually also reflected in the current approaches to COVID-19 vaccination. There are many different approaches to developing those vaccines. There are approaches of developing a live attenuated form of vaccination, where a harmless germ is told how to make pieces from another germ, and indeed similar but unrelated viruses are used to present spike proteins of COVID causing virus SARS-CoV-2. There are also approaches to develop vaccines based on inactivated forms of the virus, and lastly, there are also vaccine strategies based on delivery of subunits, more specifically the spike protein, and DNA and RNA-based approaches to COVID vaccination. As you can see here, there are many vaccines currently in development, and also some approved vaccines based on all of the strategies. In case of RNA-based vaccinations, that's the Moderna vaccine, and also the approved vaccine produced by Pfizer and BioNTech. In contrast, the AstraZeneca and University of Oxford vaccine is based on a viral vector, where a different backbone is used to deliver a part of the COVID-19 causing virus. And there are also other approaches used by Chinese companies Sinovac and Sinopharm. Here, an inactivated form of coronavirus is used. In terms of drawbacks and advantages, those vaccines are very different. In case of inactivated vaccines, those are not as effective as live viruses, and they likely need to require a booster later on. So again, all of those vaccination strategies are highly promising, and I'm personally convinced that we are going to be able to deal with the virus at a later time point this year, if the vaccination efforts remain as strong as they currently are. So fingers crossed. So what happens if the immune system goes wrong? If the immune system, both innate immune system and adaptive immune system, go wrong, this leads to development of hypersensitivity reactions. It can also lead to development of immunodeficiency. So In case of hypersensitivity, the system suffers from excessive undesirable reaction by the immune system to harmless allergens. It causes cell damage through excessive immune response to antigens that are actually harmless. It can be due to overreaction to infectious agents, but also overreaction to environmental substances. Lastly, hypersensitivity can be developed to drugs as well. Therefore, it is extremely important that you understand how it works. Lastly, hypersensitivity can be due to overreaction to self, and in this case we are talking about autoimmunity. In case of immunodeficiency, we are looking at loss or inadequate function of various components of the immune system, and this can be caused by any part or state of the immune system, for example by failure of physical barriers, phagocytes, B lymphocytes, T lymphocytes, complement, or natural killer cells. As a result of immunodeficiency, the immunocompromised host has an impaired function of the immune system, and it's a really high risk of infection. And this is the case. In several genetic disorders we have looked at earlier this term. But we are going to be having a closer look at many of those immunodeficiencies in our lecture dealing with gene therapy later this term. So those disorders have often genetic causes, like the SCID disease, but can also be acquired, as in case of AIDS. Can be acquired also due to malnutrition, leukemia, but last but not least due to pharmacological intervention. And sometimes this pharmacological intervention is actually made on purpose. One example of actually intended reduction of the immune system is in case of transplantation of organs, where I actually do want to reduce the level of immune response to avoid rejection of the transplanted organ. Because again, as you should remember, all of our cells do present proteins produced inside of the cell on the surface in MHC molecules. And if the immune system of the recipient or host is still active, this would lead to rejection of the organ. So, let us summarize what we have learned about the adaptive immune system today. First of all, the adaptive immunity develops following an entry of the pathogen into the host. It comes into action after innate immunity fails to get rid of a microbe or pathogen. It has a memory to deal with subsequent exposure, and this is different to the difference to the innate immune system, and it happens through specific cells, namely T cells and B cells. Adaptive immune response is also the basis of all vaccination strategies, although the approaches to vaccination can be slightly different. The failure of the immune response can result in hypersensitivity and immunodeficiency. And this is all I wanted to tell you about the immune system at this stage, and you will hear much more about immunity in part 3 in a lecture series delivered by Dr. Edwards. Thank you very much for your attention, and I'll see you soon. Bye.